Have you ever walked by a river and smelled that distinct fishy scent in the air? It turns out that smell might be more than just a smell. It could be a clue, a tiny breadcrumb left behind by the animals living in the water. Scientists have discovered a groundbreaking new way to track salmon, and it doesn't involve catching them or even seeing them. Instead, they are listening to the whispers of life carried on the wind. They are collecting DNA directly from the air. This might sound like something out of a science fiction movie, but it's a real and exciting development in the world of biology. It opens up a whole new chapter in how we can study the natural world around us. This incredible technique relies on something called environmental DNA, or eDNA. Think of it like a trail of glitter that every living thing leaves behind. Animals are constantly shedding tiny bits of themselves into their environment. This could be skin cells, scales, waste, or even just mucus. All of these little pieces contain the animal's unique genetic code, its DNA. For years, scientists have been brilliant detectives collecting this eDNA from water and soil samples. By analysing the DNA found in a cup of river water, they can figure out which fish, amphibians, or insects live there without ever having to disturb them. This method has completely changed how we understand biodiversity. But what if you couldn't get to the water? What if the river was too dangerous or too remote to access? A team of researchers from the University of Washington asked a bold question. If DNA is in the water, could it also be in the air right above it? The idea is that as water churns and sprays into the air, tiny droplets called aerosols get kicked up. These aerosols could be carrying the very same eDNA that scientists find in water samples. If they could capture these airborne particles, they could potentially identify the species living below just by sampling the air. This was a huge leap of faith, taking a trusted method and applying it in a totally new and untested way. This concept is so exciting because it pushes the boundaries of what we thought was possible. We often think of DNA as something you need to get directly from an organism, like with a blood sample or a cheek swab. Then we learned we could find it in the environment, like a footprint left in the mud. Now, we are learning that these biological clues can float, that they can be carried on the breeze. It suggests that the air around us is like a library, filled with invisible stories about the life happening all around. All we need is the right tool to read those stories, and that's exactly what these scientists set out to build and test. To test their idea, the research team set up a fascinating experiment right next to a river. They needed a way to catch those tiny invisible particles floating in the air. Their solution was simple but clever. They used a device that was basically a specialized vacuum cleaner. This machine was equipped with very fine filters designed to trap microscopic particles as air was pulled through them. They placed these air sampling devices at different spots along the riverbank, letting them run for a set amount of time. The hope was that as the river flowed and bubbled, it would release a fine mist of water droplets carrying salmon DNA into the air, which would then get stuck on their filters. The team didn't just place their samplers anywhere. They chose their locations carefully, positioning them near a fish ladder where migrating salmon were known to be very active. This increased their chances of detecting a strong DNA signal. The process was patient and methodical. After the air filters had run for a while, the scientists carefully removed them and took them back to the laboratory. In the lab, the real detective work began. They had to extract whatever tiny fragments of DNA were trapped on the filter paper. It's a delicate process, like trying to find a single needle in a massive haystack, but with advanced genetic tools it's entirely possible. Once they had the extracted material, they used a powerful technique called qPCR, which is a way to find and copy a specific piece of DNA. In this case, they were hunting for a genetic sequence that is unique to salmon. It's like having a special magnet that only sticks to salmon DNA. If salmon DNA was present in their sample, the qPCR machine would make millions of copies of it until there was enough to be detected. The anticipation must have been huge. Would their filters be empty, Or would they find the proof they were looking for? The results came back, and they were a resounding success. They found salmon DNA. This confirmation was a huge moment for the research team and for conservation science. It proved that, yes, you can detect the DNA of an aquatic animal from the air. The invisible had been made visible. They didn't just find a little bit of DNA. They found a clear and strong signal, showing that this wasn't just a fluke. Their experiment demonstrated that the air above a river is indeed carrying a detectable signature of the life within it. This successful test was the first step toward developing a completely new method for monitoring wildlife, one that could take scientists to places they've never been able to study before. Traditionally, counting fish populations has been a very hands-on, labor-intensive job. For decades, scientists and conservationists have relied on methods like physically standing in a river and counting the fish as they swim by. They also use tools like sonar cameras placed underwater, or even conduct surveys from helicopters. While these methods are valuable and have given us a lot of important data, they all have their limitations. They can be expensive, time-consuming, and sometimes stressful for the fish. Imagine trying to count thousands of salmon moving quickly in murky water. It's easy to miss some, or count others twice. The air DNA method offers a powerful alternative and a great point of comparison. While traditional methods count individual fish, eDNA sampling tells you about the presence of a species. The air sampling results from the University of Washington study, lined up beautifully with the data collected from more traditional methods. When the fish counters and underwater cameras showed more salmon in the river, the air filters collected more salmon DNA. When the fish numbers dropped, the DNA signal in the air also became weaker. This shows that air DNA isn't just a simple yes or no for presence. It might also give us a rough idea of how many animals are there. One of the biggest advantages of this new technique is its potential for use in remote or hard-to-reach locations. Think about a steep, rocky canyon or a dense, inaccessible forest where a river flows through. It might be impossible or extremely dangerous for a team of scientists to set up equipment or spend days counting fish there. However, it might be much easier to place a few automated air samplers, or even use a drone equipped with a sampler to fly over the area. This could give us our very first glimpse into the biodiversity of these untouched ecosystems, helping us find and protect hidden populations of fish and other animals. Of course, the air DNA method is not perfect and has its own set of challenges. The scientists found that weather could play a big role. For example, rain could wash the DNA particles out of the air, making them harder to detect. The direction and speed of the wind could also affect where the DNA travels and how concentrated it is. Choosing the right kind of filter and the best location for the sampler also makes a huge difference. Scientists are still working to understand all these variables so they can make the method as reliable as possible. It's a new tool, and like any new tool, we are still learning how to use it best. The success of this salmon study is just the beginning. It opens up an entire world of possibilities for ecological research. If we can detect salmon DNA from the air, what other aquatic species could we find? Scientists are excited to see if this method could be used to track other important fish, like sturgeon, or even more elusive animals like freshwater mussels or aquatic insects. It could be especially useful for finding and monitoring endangered species. Imagine being able to confirm the presence of a rare, threatened frog in a remote wetland without ever needing to disturb its fragile habitat. This non-invasive approach is a huge win for conservation. The technology could also be expanded beyond rivers and streams. Could we use air samplers over oceans to track the migration patterns of whales or dolphins as they surface to breathe? Could we monitor the health of coral reefs by analysing the DNA floating in the air just above the water? These questions are no longer just speculation. They are the next logical steps for researchers to explore. Each environment will present its own unique challenges, but the fundamental principle remains the same. Life leaves a trace, and we are getting better and better at reading those traces, even when they are floating on the wind. This innovation is a powerful reminder of how technology can help us connect with the natural world in new and surprising ways. As our tools for genetic analysis become more powerful and more portable, we may one day have handheld devices that can give us a real-time snapshot of the biodiversity in the air around us. This could empower not just scientists, but also local communities and citizen scientists to participate in monitoring and protecting their local ecosystems. It's about making science more accessible and allowing more people to become stewards of the environment. Ultimately, the story of tracking salmon with eDNA is one of curiosity and hope. It shows that there are always new mysteries to solve and new ways of looking at the world. By thinking creatively and pushing the boundaries of science, we can develop incredible new tools to help us understand and protect the amazing diversity of life on our planet. The answers to some of nature's biggest questions might not be hidden in the depths of the ocean or in the darkest parts of the forest. They might just be floating right in front of us, carried on the air we breathe.